So my topic today entitled Omics and Biomedical Applications. But in this case, I will specifically on the 3D cholangiocarcinoma model. So I think Dr. Apinya and Dr. Jutarop has a very well introduction of cholangiocarcinoma already, but I also have different pictures. Okay, so the cause and the risk of cholangiocarcinoma in the northeastern region and also the north region of Thailand occur from eating raw fish. The fish, not all the fish, but a freshwater fish that live in this pond, swamp, or the river that the villagers catch in the nature, and then they make the food out of that fish raw and eat it. So in the food, namely, I'm not sure whether you have eaten this sort of food like koi pla, also plara that contain in the special, papaya salad, which is the very famous dish of the northeastern region of Thailand. So if you don't cook it well, that will contain a metasaccharide, which will go into your digestive system, and then it will hatch and being an adult. This parasite we call Opistrachus viverrini, or we can call it shortly like OV. So the OV will go into the bile duct in the liver and then live there and feed by the bile. And as you can see here on the right picture, so that's the cross-section of the parasite actually inside the bile duct. Sorry. Through many years, okay, the symptoms and everything progress slowly, namely like 15 to 20 years until it gets developed into a, cancer. That could be so many types of, several types of cholangiocarcinoma, including the mass-forming types here, which the cancer cells will grow at the very end of the small bile duct or inside the liver. And also the periductal infiltrating type, which is the cancer cells will grow around the bile duct, and the intraductal growth type, which the cancer cell will grow inside and block the bile duct completely. So this is the video that Dr. Jutarop has shown already. So these are the parasite actually in the video, and also in the bile duct. I don't have to show this because it's the same video as previously. So what happened next when the patient comes to the hospital and then was diagnosed with CCA? That could be an option. So that option is that CCA patient can be resectable or go through the surgical process. So if yes, they can proceed with the surgery, and then the doctor will give an adjuvant chemotherapy for 6 months with capecitabine recommended with international guidelines. But if not, there will be a palliative care with the first line is gemcitabine plus cisplatin, or the second line is FOLFOX. So what I could use my background or my expertise on this one. I am interested in creating a 3D model out of the cells. So when we talk about cancer testing preclinically, we usually look into different models. I think most of the models that you use in vitro is the 2D or two-dimensional culture of the cells, especially the cell lines that has been established during the process. But more advancing, recently they have some kind of another 3D model to be added up apart from the 2D model, namely 3D multicellular spheroid. And also the organoids and the assembloid, which will contain a multiphenotypic of cells in the microtissue. And then at the end, you have xenografts, which is in vivo study. So these platforms, usually, they are chosen, both or three of them, to validate the whole process, from the mechanisms to the drug penetration or interaction of the cells to each other. And at the end, they go through narrow down and go through the in vivo studies to see the effect of the drug, for example, with the multiple organs, and also the cancer mass that has been grafted onto the mouse or the rat. These model has different advantages or limitations. For example, if you consider a cost and time or the ease of maintenance, the 2D culture will be the most effective for that regard. However, if you are looking for more complex microenvironment or the heterogeneity, you probably move to the organoid system. But at the end, the xenograph or the in vivo study will be the most that we could go through before going into the clinical trial. So these are how we develop a 3D model or 3D cholangiocarcinoma model in the lab. So we use the plate and coat it with agarose, like very basic things that you can find in the lab, because if you're doing molecular, you run DNA, you can find the agarose gel in your lab. We coat it with agarose to make the surface to be non-adhesive. After that, what happens next after coating? So this is the comparison between the normal U-shape U-bottom plate. You can see that if you see the cell will grow in a monolar manner. However, if you coat it with agarose, there's going to be a low binding surface, and the cells tends to be more aggregate together to form a spheroid. And you can see here, this is very small comparing to my thumb over here. All right, so this method we have published in scientific report just last year. So you can find the details more about this in this paper. What we are interested in is that why don't we try with cholangiocarcinoma cell line first to make a 3D out of this method? So we have chosen 5 cell lines at first. MMNK1 is supposed to be like a normal control of cholangiocyte cell lines. And then the cell line that starts with KKU and running in number is 4 of them are all cholangiocarcinoma cell lines, but with different phenotypic features. For example, the poorly differentiated, well differentiated phenotypes. These are the results after 3D formation of these cell lines. So you can see that this is the 2D picture of all cells. And after you see the cell onto the pre-coated plate, you can see that the cells tend to be aggregate together. But that's because of the nature of the U-bottom plate already. But if you were able to make sure that the cell is actually aggregate, you have to transfer it back to flat surface and see how it goes, whether it's actually, the cell binds together or not. So that's why I transfer the cells back into the flat surface, and you can see that for KKU 055 and KKU 100, the cells just separate. Okay. However, the MNK1 and KKU 213C and KKU 213A remains the spherical shape. So that's why we keep continuing with just these 3 cell lines for subsequent experiment. After validating with Western blotting with different markers, including cell-to-cell adhesion with different types of catechorin families, the mesenchymal markers, glycolytic pathway, T-cell cycle, and angiogenesis, you can see that in the, this is a 3D of different cell lines. So you can see that some of them didn't express any cell adhesion molecules at all. So that confirms the previous results of KKU 055 and 100 that they don't binds together to form a 3D structure. So, but more expression of the cell adhesion molecule happen with MNK1, KQ213C, and KQ213A. And also, the mesenchymal markers tends to be lower. Upregulation of GLUT1, GAPDH, RDHA, glycolytic pathway also increase. Reduce in the thing like IDH1, IDH2 involved in TCA cycle. Some of the cell line increase markedly the angiogenesis factors like VEGF-C. So, also I have evaluated tumor microenvironment using hypoxia and oxidative stress probes. So you can see as in the red color is hypoxia probes, the green color is oxidative stress probe, and that is to validate the whole cell clump here. So you can see, if you look at this panel, you can see the outermost layer contains just only blue color, which means the outer layer didn't affect by the hypoxia and oxidative stress, while the inner core, you can see more signal of the hypoxia and oxidative stress. So that confirms the tumor microenvironment of the 3D spheroid that usually have the hypoxic condition and oxidative stress at the inner core of the spheroids. And it is one of the hallmarks that it will involve in the glycolysis pathway as well. So that's why I, with the collaboration of with the Phenome Center here, with Dr. Jutarop as well, we try to validate or the profile of the metabolites because it changes and happens that fast when we comparing to the 2D culture and the 3D culture. So that's why we have a look at the metabolic profile first. Using after sample collection, we do the sample preparation, and then we use UHPLC mass spec to investigate that, and then we do data analysis and try to correlate with the pathway. This is how a PSA score plot look like. So you can see with the ESI positive and ESI negative mode. So you can see that the same cell type clustered together, but the 2D and 3D, you can still see some difference in both ESI positive and ESI negative. The cluster analysis also show the alterations of the expression of the metabolites here. Oh, sorry, the box didn't come. So this is supposed to be a 2D side. If you see here, is that half of this is the 2D of all cells with with triplicates, and then the other side is the 3D. So you can see the shift or the alteration of the metabolite patterns. The pathway enrichment analysis I didn't show you here, but I mapped the details with pathway enrichment analysis with all the protein studies and everything, and try to make a summarized diagram here. So you can see for 2D cell culture, they usually have signature metabolites that involve in the biosynthesis of the nucleotide and nucleic acids, because the cells are grown in a monolayer and they proliferate very fast. However, comparing to 3D, I myself observed that the cell will grow slowly as well. And also, the metabolite involved in the glutathione metabolism pathway will be reduced, maybe to cope with the hypoxia and oxidative stress that happens during 3D formations. And also, there will be a reduction of the, let's say, the TCA cycle metabolites, and then increase in many amino acids, maybe to supply the precursors to the TCA cycle, because the TCA cycle relies on the oxygen. So after having low oxygen, so it tries to cope with the TCA cycle, and then the urea cycle increases as well. The most important thing is we have looked at the very consensus of the higher amount of the lactate appear in the 3D model, and also the upregulated of enzyme from the Western blotting results of LDHA. So that's why we shows GSK 2837808A, quite a long name, is actually a lactate dehydrogenase A and B inhibitor to prove this hypothesis whether it has like more effect on that. This is some results tested on KKU213C, which is one of the cholangiocarcinoma cell lines. So you can see that after we apply the concentration of drug, namely gemcitabine, cisplatin, and LDH inhibitor at the IC50 level of the 2D. Okay, so we have assessed the concentration before in 2D, but we apply the same concentration and see how it goes for 3D. This graph shows the typical pattern that you can see or you can observe more resistance to the 3D model, because it has another factor, because once it forms the 3D structure, there will be a factor of drug penetration to kill all the cells. When comparing to the 2D culture, after you apply the drug, every cell is supposed to get the drug out of that, but the 3D is not. So that's why it tends to be more resistance. But after you try to combine like gemcitabine, cisplatin, you can have like increasing, but just slightly increase in the effective of the cytotoxicity in the red color here, comparing to the live cell. However, if you combine, do a tri-combinatory with gemcitabine, cisplatin, and LDH inhibitor, you can restore the cytotoxicity closely back to the IC50, comparing to the 2D, which is more effective on that. So this one can be applied for drug screening in the future for cholangiocarcinoma patient. So our spheroid has mimicked the tumor microenvironment, including the gradients of the nutrient pH and the oxygen with complying with the studies that has mentioned about the 3D structure of the tumor. So we think about moving towards the multi-phenotypic cells because, as I mentioned previously, it's just the single phenotype cell lines. So why not try with the real cell from the patient? That comes to the establishment of the CCA organoid using tissue engineering technique. So we collect ethically with the tissue from the patients is the leftover specimen that the tissue needs to be verified with pathologists already. We just cut little pieces and then it will be transferred to the lab. And then we process it with antibiotic and antimycotic to kill all the germs. And then we isolate in the hood in a sterile condition. And we did the culture in the CO2 incubator. So this scheme is the explant culture, which I will show you in the next slide. The explant culture is you just chop into the small piece of the tumors, and then you put it in the cell culture medium and in the flask, and then after like few weeks, you can see the monolayer cells grow out of that piece of the explant culture. After that, you can harvest. Ideally, you can trypsinize and harvest to make a cryostock and also reassembly into the organoid using the agarose-coated plate scheme. So this is the picture of the tumor explant, but you can see already on day 7, it has some extra structure growing out of the explant tissue and some of the single cell growing here in a monolayer manner. And after 14 days, these two pictures are the same pieces of tumor, but imaged at different places on the surface. So you can see already, even the same pieces of the cancer tissue, you can see, judging from the cell phenotypes, there are multiple phenotypics already. If you can harvest and recombine it back to be an organoid, that would be perfect. I characterize roughly with immunostaining using CK19, which is we're supposed to stain the CCA cells, and then alpha SMA, which is it should be stained with this cancer-associated fibroblasts or CAF. So you can see that the 2D culture and this is the 3D culture still have at least two populations of the CCA and the CAF. And after you culture for several days, it starts to build extracellular matrix already. So this picture is trichrome staining. So you can see the structure of cells, and the ECM buildup will be stained in blue color. So this is an original match case of CCA tissue, and the cells of match case established to be organoid. Why do we care about this? So far, the scheme that we use, currently we have processed about 50 cases, and the success rate is about 60%. And these are the time frame, roughly, estimately, about 2 weeks of explant culture, and then we do another week of expanding more cells of the monolayer cells, and then we recombine it back to be spheroid or organoids, which provide you a lots of conditions to be tested using the drugs. And you can do cytotoxicity evaluation or even further analysis that you want. So roughly about a month, so I have checked with the surgeon as well after they have done the surgery to the patient. They let the patient rest, and about a month or 2 months after that, they will start the first regimen of chemotherapy treatment. So that would be a perfect time frame for the surgeon as well to know the profile, which drug or which combination of drug is suitable for that patient. So that serves the personalized medicine. So this is the final picture that we would like for this application. We would like to fill the gap. During evaluation of individual patients, you may have sequencing reports, metabolomic studies of patients, or transcriptomic study, all the omics, and then you can combine all the data, screen which is the possible candidates or the drug that will be suitable for this patient, confirm with the drug screening for spheroid, organoid, or even the cell line as well. And then you narrow down, and then you confirm in vivo before giving the regimen to the patient that will serve the personalized medicine. I would like to thank CASCAP, Cholangiocarcinoma Screening and Care Program, and also Cholangiocarcinoma Research Institute to provide the initial fund for my research, the help of Khon University Phenome Center for Metabolomic Analysis, and also the eAsia grant, and PMUB that provide the grant for establishing the organoid. Thank you very much.
Any question from our participants?
Thank you for the fascinating cell culture work.
Thank you.
Now I realize you've done metabolomics on the 2D and 3D, right? So you have repeat 3 cells, and then presumably there's another batch, right?
Yes.
I don't know whether you noticed there's a quite big batch variation, you know, from your heat map, right?
Yes.
Is it correct? It doesn't matter 2D or 3D. You always see the batch variation.
Yes. Between 2D and 3D, and also they have like a unique patterns comparing between cells as well, cell types, I mean.
So if it's the same cell and you culture today and then in the one next week, they're different.
That's why I have to control it to be, you know, very, very controllable environment that we grow 2D as well as 3D, and then collect and analyze.
Yeah, so you can control the batch variation very well.
Yes.
But I think from the, I don't know, the metabolomics at least, right? it shows the batch variation is big. I'm wondering, with this big batch variation, would it affect your doing the drug testing and the personalized medicine that you proposed later?
Okay, but if I use the real cell from the patient, I expect the variation may be a bit high with comparing to the cell line. So that's why I have to revalidate it in a match case in the real cell as well, and probably also comparing to the real tissue with match case patients.
Oh, thank you.
Thank you.